Okay, we will start the presentation, our last presentation of the WABIO. So, Daisy, do you want to open your camera so then the people we can see you? Thank you. Okay, so it's my pleasure to introduce to you. She As Steve Hoffman, she was supposed to be here with us today, but there was some confusion on her dates, and yeah, so she was not allowed to travel at this time. Um, but I'm anyways happy that she's here with us online, and I want to tell you a little bit about Daisy. So she's actually currently a professor of statistics at the Federal University of Paraná in Brazil. And she holds a PhD in bioinformatics and a biotechnology degree and statistics from the Federal University of Paraná as well, and the Pontifical Catholic University. Her research integrates precision medicine, network science, and machine learning to investigate complex disease mechanisms and develop individualized therapeutic strategies. Dr. Gysi previously held positions at Barabasi Lab, at the Northeastern University, at the Chaining Division of Network Medicine at Harvard Medical School, and at the U.S. Department of Veterans Affairs, with key contributions to biomarker discovery, non-coding RNA-based diagnostics, and drug repurposing for COVID-19. So it is my pleasure to have Daisy here. Thank you very much, Daisy. And please start. Let's welcome Daisy.
Thank you so much for having me here. I wish I could be there in person. Can you hear me?
Yes, we can hear you very well.
Good. Yeah. So I really wish I could be there in person, but couldn't happen this time. I'm gonna share my screen. Can you see me, right? This screen, right? Yes, good.
Yes, we can see.
So good. So I'm gonna talk today about network medicine from disease models to target discovery. So my goal is to talk about different approaches we use in network medicine and how all of them can contribute for us to identifying either novel targets or biomarker discovery so how they can be applied in helping or improving our health. So as we all know, network science, everything is connected. So we can think about people hanging out on a party. So if you talk to someone, you're gonna end up having an edge between that other person. So you can talk to them, you create this kind of interaction, right? But we can also think about these interactions in other ideas. So, for example, in social media, if I follow someone, that person also follows me back, we can create an edge. Or if I share a conversation with someone in any social media, or I have a comment, or I react to someone's post, they're also going to create different kinds of links, right? But we can also apply this idea that everything is connected in medicine. So we can start thinking big about how our organs are connected to one another. So, for example, if I have a brain signal about how my heart's gonna beat, or how I'm gonna chew on something, or how I'm gonna grab something, so I'm always sending signals that they can be uh, understood as edges between those organs, right? But I can also think about this as being symptoms from different diseases. So if I have different disorders or different phenotypes, I can also try to understand how the symptoms between those diseases they are connected between one another. But we can also think, sorry, about how genes are interconnected or interplaying uh, inside ourselves, right? So if we think about how genes are regulating one another, we can have this kind of interactions. We can also have interactions that they are based on how genes or the proteins are really binding to one another. So we can have different kinds of layers of information and we can integrate all of those, right? So the idea, right, when we are thinking about network medicine is that we can understand how those interactions, right, are happening and how those interactions can affect a particular outcome. So oftentimes we think that these interactions they can also be associated with a particular context. So I like saying that all those networks they are always specific to one particular context. So let's think about a disease so that those interactions they're going to be specific to that disease or that phenotype, right? We can also try to understand how genes are differentially co-expressed in a particular phenotype. We can also try to understand how drug and their targets can affect someone's response towards a medication, right? And we can also try to identify different treatments for emerging or neglected diseases. So let's start talking about phenotype complexity, right? So most of the diseases, they are not just the product of one single gene being mutated or one single gene malfunction, right? Most of the phenotypes that we have, they are based on a complexity of several factors of things happening all together. So for that, I'd like to use the idea of mental disorders example because I think that's really rich and very easy for us to understand, and that's gonna appear several times during this presentation. So what I have here is a network of mental disorders. So each one of the squares that we have here is one disease, and each one of the dots that we have surrounding this is one symptom. And we have a major issue when we are trying to identify a patient's diagnostic for mental disorder. Basically, because most of the symptoms they co-occur in several diseases, right? So in several disorders. So if we look into this component here, we see that we have several symptoms that they are shared across different diseases. And that also leads to two things. So the first one I already mentioned, that is we have an issue trying to pinpoint the correct diagnostic for one patient. But we also have a second issue that is the high comorbidity that we have across those disorders, right? So the same idea that happens that we have one symptom that is shared across several diseases, we also have the same idea, like one gene can be involved in several diseases, right? Or one disease is not caused by one particular gene, but several genes that they are malfunctioning together. Um, so that idea we can think first I showed you using symptoms, but we can also think about physical interactions that we have inside our proteins, right? So each one of the dots that we have in this network is one protein and they are connected to one another. So we're going to have a match between those proteins if they are connected, if they are physically binding to one another. So to this network where we have those physical interactions across proteins, we call them PPIs, right? Protein-protein interaction networks. And we have always to think and to remember that this is highly specific, right? So we have two proteins that they are binding at a certain moment of time or under certain conditions. They are not binding and they're going to be bound together forever. They're going to bind, they're going to create a product, and then they're going to release one from another, or depending on the context, right? If we have a disease, if a patient is under a disease state, that interaction might not happen. Or some disease or some proteins, they might bind and they shouldn't be binding. Probably because the protein is being expressed somewhere else, or they just have like an extra site to bind that they shouldn't. And that leads to a second idea. Inside this maps that we are talking about, that they are the PPIs, we can think about the disease module. So a disease module, oh, sorry, that's too loud, is essentially when I have the proteins that they are associated to a particular disease, they're going to be co-localized, such as in a city where we have neighborhoods, right? So we have a neighborhood within the PPI that is associated with a particular disease, right? What I want you to pay attention here is that, okay, so the genes that I'm highlighting here, they are associated with asthma. And when I dim out the lights, we can see that the genes that they are associated with asthma, they are pretty close to one another. However, we still can see some genes in the neighborhoods, right? That they are not really close to the main component that we have here. We're gonna talk about those later in the presentation. Okay, so if we have this disease modules, and within those disease modules, I can say, okay, so if I have a lot of genes that they are differentially expressed or they have some protein issues, they're colocalized in this neighborhood, right? We also can think that genes that they are associated with similar biological processes, they're going to be colocalized as well. And genes that they are associated with diseases that they are similar to one another, they also are going to express what we call the comorbidity. And we can measure the comorbidity between two diseases basically by the distance that we have across those disease modules. So if I look into diseases that they are associated with, for example, autoimmune diseases, they are going to be closer to one another than we would expect by chance, or they're closer to one another than, for example, autoimmune disease and a mental disorder, right? So good. So we can think about how can we apply all those concepts in different ideas, right? So one of the projects that we had, we were trying to understand the mental disorders genetic basis. So the issue here, I showed you previously that mental disorders, they have similar symptoms, and therefore we have a, a difficulty. So it's very hard for us to pinpoint the correct diagnostic for one patient. So, okay, so if we don't look into the symptoms, can we look into the genetic basis of those patients? Yes, maybe that can segregate. We don't know. So, turns out that if we look into the genes that they are associated with several mental disorders, we still have high comorbidity, right? So, we still have a giant overlap of the genetic basis of those disorders. So, very similar to what we find when we look into the symptoms. And very interestingly, so if we apply any clustering technique in the network that we have based on the genetics, we find that the disorders, they are going to cluster according to the SM5, which is this manual that we use for the diagnostic of mental disorders. So what can we do? Can we try to identify the similarity across those diseases using other techniques? So what we are going to do now is trying to use co-expression, which is basically the gene activity measured by the amount of microRNA produced by a particular gene. So what we do is, just 1 second. Need to pause 1 second. Thank you, sorry. So we measure the co-expression based on using the RNA-seq, and we measure the correlation between those genes. One issue that we have is that when we are using the raw correlation, that leads to spurious results. So we have to correct for that. One of the ideas that we can use to correct this is use the WTO, which is the weighted topological overlap. I'm going to discuss the method a bit later. But essentially, what we do is we correct for the amount of interactions and the expression levels that we have for each one of the genes, right? And then we can compare two or more networks, trying to identify the genes that they are differentially co-expressed, and we use CODINA for that. That's co-expression differential network analysis. Okay, so we started by trying, first of all, trying to understand if this methodology works and we are able to pinpoint genes that they would be associated to neurons. So in this case, what do we have? So we had an essay where we had this microRNA, so microRNA 1 to 4, um, and we believe that this microRNA was involved in the neurogenesis, right? So we had cells, two cell lines. So one was the wild type, and the second one where we removed this particular microRNA. We followed up those those plates, so those cell lines, for 4 days, and we compared each one of the co-expression networks that we create. So we created one network for the wild type, another one for the microRNA, for the one without the microRNA, and we compared each one of the days and try to see what was happening within those cells and what would lead to the non or the malformation of the neurons, which was what we were expecting to have at the end, right? So at each one of the days, we can see this particular zinc finger, so 789, showing up as very prominently. Um, what we did next is that we knocked out the zinc finger and we followed up the cells again. And we followed them for 14 days and we could not have any neurons. So once we removed the microRNA and also the zinc finger, we stopped having neurons at the end of the story, right? Um, so that shows that the approach of trying to identify target genes or regulatory genes using the co-expression network analysis, it it really works, right? So we can also think about network medicine in different approaches. So I'm going to talk briefly about using network medicine in the times of pandemics. So the goal of this project is trying to repurpose some drug. The idea of repurposing drugs is basically I have a drug that works or was developed to treat one particular disease, and I can use this drug to treat another disease. So sildenafil, which is Viagra, was first approved to treat coronary artery disease, hypertension, and angina pectoris. However, in the clinical trial phase 1, it started to show some side effects. So males, they started to have an unexpected erection. So the drug was repurposed also to treat erectile dysfunction, which is what we know Viagra for most of the times. So the idea is doing a similar approach, but trying to use this for COVID, right? So when COVID hit, we started to gather all the data. And in March 23rd, we already had collected basically the human PPI, which is the network that I showed in the beginning. We also had the SARS-CoV targets, and we had the drugs that came from drug banks. So all the drugs and their targets. So we tried to use 3 different approaches to identify the best potential drugs that would treat or to treat COVID, right? So we had the first approach was solely based on AI. So we had 4 pipelines that they were using at GNM. The second one, we use a network diffusion, which is a standard approach in graph theory. And then we had a third approach, which is the network proximity, which essentially calculates the distances between the targets and the drug targets and the disease targets, right? So we had all of that. We had all the potential targets, the potential drugs for all the pipelines. We combined them together and we did some experimental screenings. So later we validated this towards the experimental screenings that we had. One thing that is very interesting is that when we think about one of the pipelines alone, none of the pipelines were very, very good by their own. All the pipelines, they were really good when we combine all of them. So when we aggregated the outcomes from the three methods, then we had a very robust list that basically show us that if we combine efforts, is always better than working alone or trying to identify everything using one single method. Um, how did we do this? Right? So we tested 19 and 800 drugs against COVID. And we found out that, as I said, right, when we combined those results, the active drugs, so the drugs that they were actually good for treating the disease but not killing the cells, right? They were enriched in the final list of the pipeline. So we aggregated them. Later, once we finished that project, we decided to test uh, over the 6,000 drugs that they are in the broad library. So we tested 6,000 drugs. We found 172 of those, they were active. They were strongly active, and 217 they were weakly active. So basically, out of those 6,000 drugs in the first experiment in vitro, we found the set of drugs that they were tested over and over again in different cell lines until we reached the animal model. However, what we were interested in this is out of the drugs that they are working and the drugs that they are not, how are they working and why, right? So the first analysis that we decided to do was basically, do they target the same genes? No, most of the drugs, they are not targeting the same genes. And as we can see here, right? So if they were to target the same genes, we would have like one big gene in the middle that would combine all the targets. But no, we have basically two big models, one here and the other one there, um, that we have some targets, but none of them are enriched. Um, and most of them, they were not sharing targets. So there are several mechanisms that would lead to the response of uh, killing the virus but not killing the human, basically. That's what we were interested here, right? uh, so essentially there was not one single target, not one single gene that we could say, okay, this is what we have to target for treating COVID. Okay, so remember when I showed the asthma disease model where we had some genes in the outskirts, right? So I want to talk a little bit about the role of non-coding RNAs in network medicine. So few years back, we were working in this Nature piece where we wanted to understand the Human Genome Project impact, what we know and what changed after the Human Genome Project draft was released. Okay, so one of the things that was very interesting for us is we were expecting to identify more genes to be associated with diseases or more discovery in terms of the protein coding genes. And if we look here, what we see is on the upper part of the icebergs, we can see what is the knowledge that we had before the Human Genome Project was released, and the knowledge that we have in the bottom is what we learned after the Human Genome Project was released, right? The draft was published. And one of the things that was very striking for us is that, okay, we have some knowledge that we had before and they grew, obviously, but we also have regions where we had nothing known, and now we have a lot of information. So we started to dig what are those regions? What are those genes doing? It turns out that most of them, they were non-coding elements. So the discovery of non-coding RNAs in terms of association towards diseases and towards different phenotypes. So it started after the 2000, right? So in the 2000, they started to explode as we started to learn more about them and to pinpoint that they could also be associated with disease. They were not what they were called previously, just junk DNA, right? So we started to include those interactions inside the PPI, right? So we cannot call that PPI anymore. So we're going to call this PPI and NCI because we have non-coding interactions along with protein coding interactions. So one thing that was very interesting for us is that we have proteins and interactions we have within the proteins, right? So that alone, the pink part here would be what we have in the PPI, and the rest is the knowledge that we are aggregating. And notice here that we have a lot of information that comes from microRNAs and proteins. And the size of this is larger than actually the PPI alone, right? So we have a lot of regulations that comes from microRNAs towards proteins, but we also have plenty of information that are or regulations that are coming from non-coding or other elements, pseudocodes, or even within those other non-coding elements, right? So we wanted to evaluate the impact of those interactions also in diseases, right? So most of the times we are interested in associating uh, this with disorders or how can we treat them, how can we diagnose them. So we see that non-coding RNAs, they are highly associated with diseases. So the green and the light blue, light green colors here, they are representing pseudogenes, microRNAs, or non-coding RNAs, right? Long non-coding RNAs. And we see that they are highly associated with diseases. And we also saw in the previous slide that they are also interacting with proteins. So they are highly embedded in this network, right? And we know that non-coding RNAs are associated with regulation. So how that can also help us better understanding the disease modules. So if I look, for example, in the PPI and the rheumatoid arthritis module, right, we see that we have 302 genes here in total, right? But only 62% of them are connected. 62 is not bad, but if I start to add the non-coding elements here, I start to have 391 genes. Okay, it's expected to increase the size, but we also increase the percentage of genes that they are connected. So that rises to 88%. Um, for RA, it's not a big issue because we already have a disease model. We are already able to understand the disease only looking at the PPI. However, if I look, for example, to preeclampsia, where I have 33 genes associated to preeclampsia, but only connected, like they're connected in pairs, right? We have a percentage of this coverage of the network at 6%, which is terrible. We cannot do anything using network medicine in this space. If we start adding the non-coding directions, that rises to 136 genes and 82% are connected. So that means that if we want to start to think about diagnostic or targeted precision medicine, we can start doing this now. That was impossible before. Um, I showed and I talked a little bit about the comorbidity before. So we expect the diseases that they are similar to one another, they're going to be embedded in the same space, right? So if I look into PPI, for example, rheumatoid arthritis is only connected to Crohn's disease, right? All the others, we don't find a link. So they are not statistically significant. If I go to the PPI and NCI, I find several diseases that they are known to be comorbidity for RA, and we can still identify the relative risk that we have for those diseases. So all diseases that we identified to be associated or comorbidities for RA, we have evidence supporting them. Um, Good. So with this, we can say that, okay, so non-coding RNAs, they're important. They help us improve the idea of network medicine. What else can we think about? So remember, we always want to think about precision medicine, and precision medicine means uh, I want to have a good diagnostic for the patient, and I need to treat this patient, right? I cannot just tell you, good luck, you have this disease, and that's good for you, maybe. No, I want to say, okay, you have this disease, that's how we can treat you, right? So if I want to say I want to treat this patient, right, I have to start thinking about targets. um, so we decide to map which are the drugs and which are the targets that we have for each one of those targets. And we started believing that maybe during the Human Genome Project, we started to discover, as we discover more genes, that we also discover more targets, that we test new targets. And turns out that no. So in the bottom here, we have in the dark blue, we have drugs and that they are developed. And if a drug has a new target, we're gonna see this increasing, right? But we see that most of the drugs they started to be discovered, created and using targets that they were already used before. So after 2005, we can see that we stopped adding novel targets for drugs. So it's always new drugs, but the targets they are all the same, right? So we don't see an increase in the target discovery. Okay, but what are the clinical trials also saying about this? Are the drugs not passing through phase 4, or are those being tested in clinical trials and failing? What's happening there? So we map it on the PPI genes that they are associated with clinical trials, right? If the clinical trial was approved, right? Or if they are associated with diseases. So we have, and it's very light blue here that we are using. So we have diseases in this region that I'm surrounding. But we have the FDA approved drugs. They are mostly in this region. While the clinical trials are going in the other direction. So we see that there is this movement of clinical trials testing different regions, but it's still, we have a majority of diseases not having any kind of movement going towards them. So there are still several targets that could be tested, mainly for neglected diseases or rare diseases. So the rare disease space still lacks a lot of uh, drugs. Right. So I want to conclude this first part so we can move to different areas we can also think about and a more close application. So in conclusion, we know that the precision medicine is moving towards better patient certification, right? So we are getting better into identifying what the patient really has and how can we start navigating and treating those patients. We are also including different kinds of um, data into this universe. So, for example, we started to layer information from medical imaging or social contact or Fitbit information. All of these kind of layers are being added, and we can still use the basic network medicine approaches as another layer of information, right? So, as we can see in the patient stratification universe. So I want to talk about psychiatric disorders because in the beginning I mentioned that we have this huge symptom overlap, and I started with this, and I want to close with this kind of projects, right? So, as I mentioned, psychiatric disorders, they share many symptoms. So, for example, insomnia and irritability, they are common to several psychiatric disorders, most of them. So, if you go to the clinician and you say, "Oh, I have insomnia and I'm getting very annoyed very easily," um, that's not going to help because most of the psychiatric disorders, they're going to have those two as symptoms. So, we started a project where we wanted to really understand what did the DSM-5 tell us about the mental disorders. So DSM-5 is Diagnostic and Statistical Manual for Mental Disorders. Um, it's a book where we basically have the description of the disease, what is the diagnostic criteria, and how the doctor is gonna make this the clinician is gonna make this diagnostic, right? Um, it's a book where it classifies a mental disorder. That's essentially what happens. So it has different chapters, it has different disorders. And what we started by doing is we created a AI tool that was grabbing the title and grabbing what else the potential symptoms for each one of the diseases. And when we presented this to some clinicians, they're like, yeah, not that good. So we went back and we, we were really reading humans, reading the chapters and getting the symptoms that was specific for each one of the disorders. And we mapped it, we transformed this book into a network where we have basically for each one of the disorders all the symptoms that we have. So we have this bipartite network where we have the disease. So I'm looking here at cyclothymic disorder, right? And bipolar disorder, bipolar 2, substance abuse. And I have all the symptoms for all of them. And we had to make sure that the symptoms that we were talking about for cyclothymic disorder would be mapped to the same that we would have in substance abuse, for example. So we started looking into this networks for each one of the chapters and trying to identify, okay, which are the main symptoms that I have for each one of the chapters, right? So we want to understand the symptoms overlap within each one of the chapters. Good. So, for example, if I look here, a cyclothymic disorder and substance abuse and bipolar disorder related to another medical condition, I would have elevated mood and irritability, as I mentioned, that appears for almost everything. Good. So the first idea is, okay, we can identify the overlap that we have within each one of the chapters. And we believed that the chapters would be where the major overlaps would be. Turns out that when we put this full network together, most of the symptoms are highly interconnected across chapters. So I'm not so we started expecting that diseases they would have a high comorbidity within diseases that they're theoretically similar. They are constructed under the same chapter. Turns out that that's not the case. They are highly interconnected across chapters, which makes the diseases way more comorbid than we were expecting in the beginning. We have a second issue. It's very common that the patients can have several diseases simultaneously, right? So what you have in this network is the diseases, right, and their symptoms. Essentially, that's what we have. And oh, it's not zooming. In this region here, where I'm showing, we can see those pie charts where we can see that those symptoms are shared across multiple chapters as well, and that's what's tying this network all together. Okay, so we wanted to create a network-based approach to make the diagnostic for mental disorders. So the usual approach, it's using this KIT-5, that is a structured clinical interview for DSM-5. It takes several hours, so it depends patient, depends on the clinician, but that goes from 1 hour and a half to 4 hours and a half. So let's in on average, they take over 2 hours to do this screening. So it takes a very long time. And often the patient's tired, the clinician is tired. So what we are proposing here is a network-based approach to make the diagnostic for mental disorders. So the idea is to have questions that they are dynamic. So the next question is based on the answers that the patient already gave me. It's going to update the networks with new information and updates the probability of a patient having a specific disorder. So we did this using a synthetic data. So for each one of the disorders in the network, um, we create 1,000 patients, each one of them having from 5 to 100% of the symptoms. So we expect that if a patient has 100% of the symptoms, I'm already able to identify that that's disease that they have, right? Um, but I also expect that if I have less symptoms, uh, I would be more or less able to identify what does this patient has. So we measure the accuracy for this network-based approach, and what we find is that for the diseases that we tested, with more or less 50% of the symptoms, we are already able to identify what does the patient has with a decent accuracy. So basically, with this approach, we can already help the clinician in reducing the time between uh, the 2 hours. The idea is to have a questionnaire that's going to take 30 minutes that can help them to do a first screening on the patient, and then they later can make the final diagnostic using the appropriated tools. Okay, so we see that we have this huge overlap within the symptoms. If we look into the genetics, would we still have the same thing? As I showed you previously, yes. So if I look into the genetics, we have this high overlap. It doesn't matter how I'm classifying them. So there are several measures I can use, and all of them still give me a highly connected network. So can we use a multi-omics systems biology approach to try to understand this? Maybe, because we still see that the diseases, they are highly interconnected, right? So what we decide to do is focus on diseases with high comorbidity, only six, and look into the co-expression analysis that we would have for all of them. So I'm using bipolar disorder, autism, major depressive disorder, Alzheimer, Parkinson, and schizophrenia. So I'm going to focus on those six diseases. I'm going to collect data from as many data sets as possible, right? So we're going to try to identify novel biomarkers for those disorders, right? So that can, in the end, detangle those diseases. So we're going to use postmortem prefrontal cortex. So from the brain data from 1,800 patients, and we are using data from 55 studies. So we build the co-expression network for each one of the studies, we combine them, and we do this for each one of the diseases. So how do we do this? So for each one of the diseases, I have the data sets. So from one to as many data sets we have, we process, we build the networks, we combine that into a consensus, and I do this for each one of the diseases packages, right? Then I compare the diseases and I'm going to have the biomarkers. So in summary, what the WTO method basically does, so we are using this focus on transcription factors, right? Because we know that transcription factors, they are highly associated with the manifestation of mental disorders. So it makes sense for us to filter and focus on in this class of genes. So we construct the networks, which takes into account how many other interactions we're going to have if it corrects for that. So we are not using raw correlation, for example. And we reconstruct each one of the networks 1,000 times, and we filter for the ones where we have high confidence on the edges. Then we stack all those networks together using a consensus network. So basically, if I have 4 different networks, and all of them are telling me similar things about one particular edge, I'm going to keep that edge in. If, for example, this edge here, it appears in all the networks, very similar with the same weight and the same direction. So I'm going to say that I can trust this, right? A similar thing with this other one and this other one. But if a link is said to be upregulating another gene and in another network downregulating another gene, I cannot trust it. So I'm going to remove. So that's basically what the consensus network does. It reassigns weights based on the weight of evidence that I have, and the recalculates a p-value for those interactions. So you're going to have a very high confidence network at the end that comes from all different analysis. So now we have a very high confidence co-expression network for each one of the diseases. We can compare all those 6 diseases. We can think about as many comparisons as possible, but we're going to focus on 2 main ones. So one that is for neurodegenerative diseases, that's going to call Parkinson and Alzheimer, and I'm going to use healthy controls. And we're going to think about a second one where we have autism, bipolar disorder, schizophrenia, and major depressive disorder. And I'm going to compare them against the adults. So how I compare them, I have those networks, and I can say which links they co-appear in all the networks. So they are conserved, they're common in all the networks. I can also think about the ones that they change the size, so they are different in at least one of the networks. So for example, this is blue here and it's purple in the other ones. And I can think about links that are specific, so they only gonna exist in one of the networks. So I don't care about the common ones because basically they are conserved about in healthy patients and the patients with healthy and against the patients, right? I want to identify either the differential or specific ones. So on the neurodegenerative disorders, we find only 6% of the interactions in the brain are common, right? So we have the 94% is different, right? So and Alzheimer and controls, they share 37% of the network. So we find out of the Alzheimer's patients that we have 30 genes that they're specific to Alzheimer's patient and 58 that they were specific to Parkinson's. But we only find one gene that is common between Alzheimer and Parkinson. That was one cut. And this gene is associated to neuron development and differentiation. So when we were thinking about the network in the beginning, we saw that they were highly convoluted and we have this major overlap. But when we think about the co-expression analysis now, we see that, okay, they have some overlap, it's good, but we find one gene that is different between them to the other patients, right? So we can probably start detangling those two. When we start to think about the neurodevelopmental disorder, we find that less than 1% of the network is highly conserved. 30% of the network is specific to controls. We find many genes specific to autism and to depression. Schizophrenia that in theory has way more genes described to be associated with this disease has only 21 TFs, and bipolar disorder has only 2. So we are able to detangle the diseases from one another. And instead of identifying a huge overlap across all of them, we start to find a much narrow overlap between all the disorders, right? So we've also looked back into the PPI of those networks, and what we identify is that disease modules that we identified through the codina and layered then on the PPI, they are much stronger than the disease modules that we have when we just look into the ones from GWAS associated, so the ones that we have in the beginning. So we applied a combination of different machine learning and network analysis, and we are able to identify potential biomarker for neurodegenerative disorders. We combine and compare almost 2,000 samples from different data sets, 55 data sets from several disorders, and we were able to identify biomarker for them. We also find that the transcription factor biomarkers seems to be more tightly related than when we look into the PPI alone. Obviously, this whole work was not done just by myself. There are a lot of people that was involved in this and several institutions. So I'd like to thank all my collaborators and also leave my contact to you. We are hiring for our PhDs and masters, and I'm happy to take questions.
Thank you, Daisy.
Thank you.
Questions for Daisy. Question.
Hi, Daisy. Thank you for the nice talk. I have a couple of questions. First, you said that we are discovering new genes, but not new targets. And I didn't get if it is because we are like we have like a research bias, when like we are only researching things that we already know, or it's or there are not like new targets.
So we have potentially um, explored less than 20% of the potential targets that exist. And that's basically because so it's the first idea that you shared. So we are reusing what we already know. So we have a bias. So this already works. So let's keep using what we already know and not trying to re-add new targets because we have to do some. So target discovery is quite expensive, right? So you also have to ensure that you are not going to have toxicity. You don't have several other issues when you're testing a novel target. So let's keep using the targets that we already know. But that also has a cost, right? So if you're always using the same things, you're not discovering, there is no innovation, there is no treatment for neglected or rare diseases.
Yeah, I see. Thank you. And my second question is, so you found a lot of overlap between symptoms, an example, and also I guess between genes network.
Mhm.
Uh, and I was wondering if it's enough to use like pair interactions to model your disease, or maybe if you have like three symptoms, it's different of having two of them, something like that.
Yeah, that's a very good idea. So we often think about the pairwise interactions because it's much easier for us to navigate and understand the network when you have the like this kind of like two order interactions instead of thinking about how, when you start to add like three or four symptoms that they would lead to the disease. But that's something that could be done.
Nice. Thank you so much.
Thank you.
Hi Daisy, very thanks for your talk. My question is, which program do you use to interact a PPI network with another network like NCI network?
I like in software, you mean?
Yeah, like which program?
I use R.
R?
R.
And, and well, tell me about the the benefits of use this interaction with with different networks. Or,
So you mean when we add the non-coding,
yeah,
right? So the benefit is that you have a larger network that you can pull in much more information that you're neglecting when you're using the PPI alone. So, for example, if you're thinking about preeclampsia, one of the examples that I showed, you are not able to identify any disease model. You are not able to identify potential targets. You cannot think about uh, diagnostic or anything just because you don't have a connection within the network, right? When you start to have the non-coding, you start to connect the dots, right? So that can help us to create uh, better diagnostic tools or anything, or even understand the disease mechanism. Because if you have only two genes that they are associated with one another, right? That's the case that we have for preeclampsia, for example. Like, how can I start to think about which is the pathway that is associated, or how can I understand the disease mechanism, right? When you start to layer the non-coding, you start to think, okay, this might be the disease mechanism that I have to stop, or how can I make this other mechanism or this pathway to work again.
Thank you very much.
You're welcome.
Hello, thank you for your talk. My question is, which kind of tissue do you usually use? And do you think that different diseases will have different co-expression networks depending on the tissue? If so, how would you choose the best tissue of those available?
Okay, so the PPI we often construct, the initial PPI, right? We often construct as tissue agnostic. So it's the same PPI for all tissues. But if I only want to study, for example, the brain, I'm going to focus on a PPI that is a brain fix, like a PPI that is specific for brain. So I filter the network to have a specific network for the brain. And then I reconstructed these modules and everything there. That's the first question. The second one was the co-expression, right? So for the co-expression analysis, it's very different depending on the tissue that you're using. So you have to be very careful when you are thinking about which tissue I'm going to use. So let me give you an example. So we were studying rheumatoid arthritis, right? So RA, and we had different tissues. So one of them was the synovial tissue where the disease manifests, right? One of the tissues that the disease manifests. That's the liquid that we have within the cartilage, right? So within the joints. So we have that tissue, and that was beautiful. We get a good signal. We can identify which could be the potential targets, and beautiful. If we try to find the same things in the blood, we were not able to get anything because the tissue, like in part of those genes, they were not expressed in the blood, or part of them, they're just like the signal gets diffused or mixed out. So you don't have as strong signal as you would expect. So for the co-expression analysis that I showed, we use prefrontal cortex, so brain, right? And obviously it has to be post-mortem, right? Because we cannot chop people's head open and get the brain. So you have to be really careful when you are thinking about this. So for example, the genes that we are identified, we also, if we want to do the same analysis using alive volunteers, we probably going to use blood, right? Are those genes also expressed in blood? You have to double check that, right? And how is the correlation between them with the tissue that you want to study? That is, in this case, brain.
Thank you.
You're welcome.
There is a question online. Just one person is asking something. Can you open your microphone, the person who's asking online, because we are not able to read it?
I can read it. Oh,
Can you read it? Or
yeah. Sorry,
yeah.
which is the university is hiring for masters? It's the Federal University of Paraná.
Okay.
Yeah. So
There
it's
are
in
more questions.
the computer science program. Just send me an email. So I have positions for PhD and masters.
Hi, thank you for your wonderful presentation. I was wondering if the network is made after or before mental diagnostic, like
It's after the patient's already dead.
yeah. Thank you.
We have to get the brain out.
Hello, thanks for your presentation. I am asking something that maybe is not a good question, but I will ask
Don't
it
worry.
anyway. What about drugs? Are we using the best drug for each disease, or there is also overlapping? And
Ooh.
does your research can guide medical uses of drugs, or what about it?
Yeah, okay. So are we using the right drugs for the right patients? Short answer is no. Long answer is depends. So we know that several, so for example, drugs, they are often developed for males, right? Because females, we have hormones, and oftentimes when a drug is discovered, the first clinical trials, they are often done in males, and then later they start to add more sex, like equality. But most of the drugs, they were developed for uh, theoretically white males. So to give you an idea, 70% of females or Asians and African people from heritage, they do not respond for ibuprofen and paracetamol, which are like spreadly used and everybody knows and use them when have a fever. There are people that do not respond. And that goes for most of the other drugs as well. So we have equality access bias towards drugs. So pharmas are trying now to get much better in this and trying to get different people from different backgrounds in the clinical trials to assess really how the drug is acting. But it's still we are far from having drugs for everybody and having the best drugs for each one of the diseases.
Hi Daisy, again it's me.
Hi.
About like interactions between pairs or of disease or symptoms or more like I mean not considering only pairs interaction between pairs, but more like three, four, five. I was wondering what are the technical challenges and if it is expected if we consider these interactions like high order interactions, is
Mhm.
it expected to have better diagnosis or?
I don't know. So I think that it's going to have a lot of technical difficulties because uh, identifying triplets as interactions in a bipartite network, I think that's going to have a lot of issues to really construct all of that, um, and to have a final network that is going to work. I cannot tell you
Okay,
at
thank
this
you.
stage because I haven't tried it.
Thank you.
Thank you.
Any more questions for Daisy? Okay, Daisy, thank you very much for this amazing talk and hope to see you in Mexico maybe next time.
Yes. Thank you so much for having me here. It was a pleasure.
Thank you.
Thank
Okay.
you. Bye.